Ascomycetes is another phylum in which we find yeast like Saccharomyces that are used in the manufacturing of beer, molds like Neurospora, and even different kinds of cup fungi that are illustrated, the orange peel cup fungi in the lower left-hand corner. Now, Saccharomyces, they're often very proprietary or secret species of Saccharomyces that are used in industry and manufacturing to make different kinds of food products. The Neurospora, in decades past, have been used in genetic research. Notice that the ascospores are lined up like little footballs and what look like long finger-length structures. Those little long fingers and the arrangement of the spores are very precise, and that's why Neurospora has been used many decades ago in the scientific investigation to the cause and the source of genes or DNA replication. In the early 1940s, George Beadle and Edward Tatum used various mutant strains of Neurospora to study metabolic pathways. Using previous research as a stepping stone, they were able to determine, using these various strains or types of Neurospora and the arrangement and pattern of growth of the little ascospores in these long little filaments, they were able to determine that one gene was responsible for one enzyme. And of course, it was part of the early research in trying to determine what genes were and how they functioned. Basidiomycetes is another phylum of fungi that include the mushrooms that reproduce asexually using basidiospores. There are all kinds of mushrooms, but the one we're going to focus on that really catches our attention is the genus Amanita, often called the death angel, and various other kinds of sort of scary titles. This particular mushroom produces a toxin or a variety of toxins that can make animals and people very sick if ingested, thinking that they were essentially food. And the types of symptoms that you might experience are almost flu-like symptoms. And the toxin itself, once ingested, actually inhibits protein synthesis in the cells of animals. Now, the targets and where the toxin affects protein synthesis in eukaryotic animal cells is very precise. As such, the Amanita toxins are now used, and have been used for quite a while, in the study of translation or protein synthesis in eukaryotic cells. Some other examples of Basidiomycetes are the shelf fungi, the puffballs, bird's nest fungi, and the earth stars. Notice that they all have very different kinds of morphologies or shapes, and in fact, the puffballs, if collected at the appropriate stage of growth, can even be eaten just like certain edible mushrooms. Notice also the size. That's actually a picnic table. And notice that there are two large puffballs that almost completely cover the surface of the picnic table. The phylum Deuteromycetes is also called the Fungi imperfecti. Most of the genera in this phylum do not have any obvious sexual reproductive cycle. Most of them are simply identified based on their asexual reproductive sporulating structures. Most of the medically, clinically significant fungi are found in this phylum. One example of a species in the Deuteromycetes that's of clinical significance is Coccidioides immitis. This is a pathogenic or disease-causing mold that can produce arthrospores, which are barrel-shaped or can-shaped, that can be found in the air and the soil. There are certain places in the world where this mold is essentially always found in the soil. And when the soil gets disrupted, then we see an increase in the infections caused by this mold. Coccidioides immitis is typically found in the San Joaquin Valley in California. It's always found in that area, and as such, we call it an endemic organism, because if we travel that geographic location, we will very likely encounter this particular mold and its arthrospores in the soil. Now, when inhaled, this organism causes a condition that we call valley fever or San Joaquin Valley fever. It can also cause pneumonia, and if on the skin, it causes a disseminated cutaneous mycoses that we can clearly see in the central images on the top and the bottom. Now, whether it's a skin infection, which would be the cutaneous mycoses, or a systemic infection like the pneumonia or the valley fever, notice that when growing in the tissues of the person that's infected, the coccidioides forms these little nodules. We have different magnification images of the nodules, starting at the bottom, which is a gross image growing in lung tissue, all the way up to the magnified images from those histologic sections. These little spherules are packed full of the tissue form, or sort of the yeast form, of coccidioides when growing in tissues at essentially 37 degrees Celsius. So Coccidioides immitis is an example of a dimorphic fungi, D-I-M-O-R-P-H-I-C, dimorphic. It will grow as a mold at room temperature, but grow in the form of a yeast at body temperature, roughly 37 degrees Celsius. The little spherule is an example of coccidioides growing in the tissue. Those dark purple structures growing and collecting around the spherule are white blood cells that are trying to defend you from the infection from the mold. Here's a large mass. It's a macroscopic piece or spherule in liver tissue. So you can see they get quite sizable. Now, we typically identify the different Deuteromycetes by their macroscopic colony morphologies and the microscopic conidia and any other kinds of microscopic structures. If they're tiny, we call them microconidia. If they're large and clove-shaped, we call them macroconidia. We'll also find various kinds of spirals and all kinds of other filamentous shapes that help us in the clinical laboratory figure out which particular kind of Deuteromycetes are we working with.